Hi, and welcome to our gathering on the cerebellum. Today, I am very happy to have with us Aleksandra Badura. Aleksandra is going to be talking about the work that she had done when she was a postdoc.
Somebody on the Zoom was from Cornell, and you can see.
And I'm going to give you a chance to listen to it, but I want to first introduce her. So Aleksandra received her PhD in neuroscience at Erasmus, training with Chris, where she studied the impact of afferent inputs on cerebellar activity and motor coordination. Then in 2012, she moved to Princeton here in the United States to join the laboratory of Sam Wang. There, she discovered that granule cells acquire signals predictive of motor performance. And then 2015, 10 years ago, she returned to Netherlands where her research is focused on the role of cerebellum in autism. And she's making a wonderful headway in understanding the neural basis of autism and its regard to cerebellum, cerebellar involvement in development, as well as abnormal cognitive and social behaviors. She's now an associate professor at Erasmus, and we're going to hear about her work on non-human primates. Good to have you, Aleksandra.
Thank you. Thank you so much for having me. Welcome, everyone. Maybe it might be a little bit surprising given the introduction that Reza did and the fact that my lab indeed studies neural mechanisms that underlie autism. Why I chose to talk about cerebellar activity during voluntary eye movements, particularly in non-human primates. However, I hope that in the course of this talk, you will see the link and the reason why these types of fundamental studies actually give us a lot of insights into conditions like autism that we further can disentangle in humans and in mice. So for just for reference, I wanted to tell you that not just if you look at the CDC numbers, the autism prevalence rates are high, not only in the US, but also in Europe. In the data from the Statistics Bureau in the Netherlands shows that in 2025, around 3% of the Dutch population is autistic. And you can see this is actually provinces. I really wanted to show this map that even though the Netherlands is very small, we have distinct provinces. And you can see that in some of the provinces, there's around 500,000 people. And this is actually the number of people who corresponds to the autistic population. And when I talked to autistic individuals, but moreover, when there was a survey that was done at over 2,000 people with autism diagnosis, one of the most prevalent questions that they have is, why does my brain work differently? What's happening in my brain that maybe my behavior is a little bit different from everyone else? So when it comes to this behavior, currently autism is diagnosed based on these core differences, core characteristics, and these are communication and social differences. I know it says deficits. However, you have to also understand that sometimes this behavior might be just different and something that we are not used to. So also hyper sociability might here play a role, and repetitive and not flexible behaviors. And it's very interesting because autism has many different underlying causes. We know that there's a very, very strong genetic component. But nevertheless, regardless of the etiology, these types of core mechanisms, together with also sensory motor deficits and sensory motor delays that are observed, particularly in childhood, are prevalent across most of the individuals who are autistic. And I think for this audience, I don't really need to convince you, but I think that for a lot of people who do not study the cerebellum, but study autism or other part of the brain, this finding was very surprising, that cerebellar injury around birth, cerebellar defects around birth actually carry a high risk for autism. And this is only comparable to that of having an autistic twin. So in other words, next to the genetic causes, the cerebellar insult is right there when it comes to the risk factors. This has also been already known for over 20 years, that if we look at the cerebella of people with autism diagnosis, we often see changes, so things like hypoplasia of the cerebellum. And I'm happy to talk about it offline, and I hope that maybe in a year or two I'll be invited back to talk about work that I'm now doing on human cerebellum, when we're really looking at these cerebellocortical developmental trajectories. And we also see that actually the delay in cerebellar development can impact the development of the neocortex. And we know it from beautiful work of, for example, the lab of Peter Strick, that there are these reciprocal connections between the cerebellum, not just to the motor or sensory cortex, but also to the forebrain areas that have to do with higher cognitive functions. And when we looked at the mouse models that we studied, we saw that actually those are mice models where there are cerebellar mouse models of autism. What I mean by that is these mice have a loss of function mutations in the cerebellum, or they have inactivation of parts of the cerebellar network. And we can see that actually a lot of them have autism-like deficits in the behaviors. And also, if you see, for example, motor coordination is very consistently affected in these mice models. So is the motor learning. But for example, cerebellar firing patterns, we find both that they can be increased and decreased. And this is a little bit already of a spoiler of the things that we find in a dentate, because the cerebellum is not simple. So you can find increases in the firing pattern, you can find decreases in the firing pattern, and both of them might lead to pathological outcomes. And I wanted to highlight that this cerebellar influence on autism has been replicated across many labs. So there's a paper from when I was in Sam's lab that shows that when you lesion with dreads the cerebellum, particularly the lateral cerebellum, you have this loss of sociability, as seen here, where the mice do not choose between a novel mouse and a novel object. This was also very apparent in the paper from Peter Tsai, where Catherine Studley is the first author, where they also showed this, and also another paper, or several papers from Peter's lab, together also with Mustafa Sahin, that shows that if you delete autism risk genes in the cerebellum alone, you get these deficits. And already here we could see that there is something very peculiar about the lateral cerebellum, because there's more and more reports about the importance of the lateral cerebellum. So the work from Jessica Verpute's lab, who you will hear from, so I'm not going to spoil it too much because you're going to hear from her later this year. But she also shows, and her lab showed, that both this lateral cerebellum and the dentate nucleus is very highly involved in these autism-like behaviors. I say autism-like when I talk about mice, because of course the one-on-one translation between the mouse behavior and the human behavior is somewhat lacking. So I use the term autism-like. And particularly the paper from Catherine from 2018, I think this was a very nice study, because it showed that the pattern of right hemispheric cerebellum to left neocortical regions, cognitive neocortical regions, is altered in both humans. So you can see here, this is the, can you see my cursor? Yes, okay. So the right CRUS1 to the left parietal lobe. This was increased in the autism group, and they also showed this pattern here in these mice mutants. So there is something specific that we think happens in this lateral part of the cerebellum and the hemispheric cerebellum. And actually, this was something that I did earlier this year. I was just wondering how much literature there is. This is not a systematic review, so probably there are duplicates. This number of papers is probably going to be smaller, but this just shows you how much this field, how much interest there is in the field. And there's many papers that have been written about this. And also it gathered interest in the in the layman media. So this is not just something that we talk about in the lab, but if you see here NPR, I didn't include it, but even Dutch media sometimes covers these findings on cerebellum and autism. And there's now also interest in the modulation of the cerebellar network to influence behavioral deficits. So if we look specifically at this lateral cerebellum, so already in the work from Buckner et al., where they mapped, this is now the human cerebellum, and what you're looking at is like this unfurled. So if you have the cerebellum is very, very folded. And if you think of unfurling this 3D structure into a 2D picture, this is the picture of the human cerebellum. And we can see that particularly these lateral regions are connected to the frontoparietal network and default, something that we call default mode network, that is very important for cognitive processes. This was a paper from 2011, but I just want to show you again, I love replication. So whenever there's a paper that comes and shows something that happens in the CRUS-1 that has to do with autism-like deficits or in dentate, I'm very, very happy. And I'm sure that people who do human work are equally happy to see this type of replication. Because we also saw that when it comes with Guel et al. 2018 paper, we see that there are language and default mode network. So DMN is the default mode network. You have it in the lateral part of the cerebellum, which sort of corresponds to CRUS-1, CRUS-2. And you also have these working memory and frontoparietal network areas. And these are also hemispheric regions, but very, very lateral. So this is hemisphere more in the middle and the hemisphere, very lateral parts of the hemisphere. And finally, we actually now are slowly departing from the work, from looking at the cerebellum purely from anatomical division. So I was telling you about CRUS-1 and CRUS-2, and that has to do with the lobules. So you see these black dotted lines that denote these anatomical divisions. But nowadays, particularly when it comes to the human cerebellum, we use functional maps. And here in these functional maps, you see that these lateral regions are things to do with divided attention, word comprehension, visual letter recognition, and also divided attention and emotional processing. So this is what we see in the lateral cerebellum in the human. In this, all of these studies are fMRI studies. And I think this is particularly of interest to us because, of course, things like divided attention, language processing, emotional processing, these are some of these processes, some of these behavioral patterns that are affected in autism spectrum disorder. And I have to say that when I was going back and looking at who actually did the very first recordings in the lateral cerebellum, who was it? I have to say that to my delight, maybe correct me if I'm wrong, maybe you find a different, an earlier study, but I found this study from Ron and Robinson from 1973, where they actually electrically stimulated the lateral cerebellum, and they evoked eye movements. And I think that this is quite interesting to everyone who's at Johns Hopkins, because I found out that David Robinson was one of the founding members of the Department of Biomedical Engineering. And so this to me was, when I found it, I was really being like, oh, wow, this is like completely full circle. There were also later papers that showed that electrophysiological recordings from these regions, and this is aligned to saccade. So SC here stands for saccade, and they showed that the cerebellum, this lateral part of the cerebellum, is active when the monkey, so these are all monkey recordings, not human primates, rhesus monkeys, when they make an eye movement. And when you lesion this part of the cerebellum, the monkeys can still make a saccade, But as you can see, the timing and the position of the saccade is way more variable. This is important because traditionally, if you think of eye movements, like purely eye movements, things like we study oculomotor reflexes, vestibular ocular reflexes, we normally talk about vestibulo-ocular cerebellum, and this is located either in the flocculus paraflocculus or in the posterior part of the vermis. And when it comes to the monkeys, actually vermis and the vermal regions are mostly recorded for these types of tasks. But you can see that there's something specific here. So it can evoke eye movements, but when you lesion it, it's not like you abolish eye movements. There's something else that these regions are doing. And if you look at the retrograde stainings, antrograde, retrograde stainings, it is true that these antrograde and latrograde staining from CRUS1 and from CRUS2 connect to not only the motor cortices, but also to the MPFC. But we do not have a direct visual cortex to cerebellum pathway, so that is something to keep in mind. So there's been this ongoing question whether this lateral cerebellum is involved in the decision rather than execution. So we know this vestibular cerebellum, if you affect this one, if you lesion that, then the saccades become really unreliable. In the mouse, if you lesion the vermins, you can also have a lot of ataxia. But when it comes to the decision what to look at and where to look at, and this is some work that I'm going to show you from Tanaka's lab. This is something that also we were very inspired when we started recording from our non-human primates. This is a very simple task where the monkey is asked to make a pro-saccade, so saccade to a target, or when the target appears, the monkey is asked to is rewarded for making an anti-saccade. Anti-saccades have larger delays and are also more cognitively demanding because you have to suppress first this reflex of going to the position of the initial stimulus, and then you have to plan where your eyes are going to land and then make the eye movements. So this is a more cognitively involved task. And in fact, we also use the anti and pro-saccade tasks in humans. And we also see that there's, for example, a strong deficit in the anti-saccades for kids who are prematurely born, for example, or who also have neurodevelopmental deficits. So what they did is they recorded from dentate nucleus, and so did we. So the rest of the talk is going to leave the cortex and focus on the dentate. And I will tell you why. It's simply because we actually started recording from the surface of the cerebellum, so from the cerebellar cortex. And then we realized that we're never, ever going to get anywhere because the heterogeneity that we saw in the cortex was astonishing. And back then, all these recordings were done with tungsten electrodes. So you get one cell per time. I mean, you can go a little bit lower, but it's very time consuming. So we decided, and back then, a lot of people have decided to focus on the dentate because this is the connection. So the lateral cerebellum projects to dentate, and then the dentate is the ultimate output of the cerebellum. So a lot of people looked at the dentate recordings just like here, and you can see that actually these neurons in the dentate are very active in this instruction period. So when the monkey is instructed where to go, and that the activity is higher for this higher cognitively demanding task. And then another work from his lab showed that when you do this synchronized or reactive saccades, very short synchronized is like when the monkey knows that if there's a probe coming here, there's a stimulus coming here, then the next one is going to appear here with a given delay. So there's it's really synchronized because like stimulus, saccade, stimulus, saccade, stimulus, saccade. Or a reactive one where basically the monkey doesn't know where the stimulus is going to occur and it just has to react and it's rewarded for making it. They showed that first of all, a third of the recorded neurons were active actually regardless of the saccade direction, but they really showed this greater activity for the synchronized saccades and particularly in this predictive time. So not when the saccade occurred, but before that. And this is, keep in mind, this is this instruction period. So as if when, you know, the monkey is gearing up, like, okay, I'm going to make an eye movement. I'm going to make an eye movement in the moment. And where do I have to keep do the eye movement to? So we went a little bit further because what I was also really interested in, and also Chris at the time when he set up his monkey lab in the Netherlands, we were also really interested in reward encoding in the cerebellum. And for reward encoding, you also need error trials because, of course, you need to sometimes not get a reward, and sometimes you want to have like things that fail. And I was very interested in cognitively demanding tasks. So probably a lot of you have had this discussions that we try to think about what does the cerebellum do for complex movements. You know, we talk about playing the piano, playing the violin, and then we study like a pro saccade, which is very valid and very important to our understanding of what does the cerebellum do. But this jump from having a very complex task to studying a very simple types of behavior sometimes creates a gap in our knowledge. So we decided to do recordings of this visual attention task called covered visual attention. And I just wanted to tell you what it is. So imagine that you're presenting at SFN or at FENS and you're standing in front of Reza. So you're very, very focused and, you know, you're talking just about your work to Reza, but you've been talking for 4 hours and you're also very, very hungry. And what happens is that someone passes in the corner of your eye and they're having a very juicy hot dog. And then what happens is your eyes are completely locked on Reza, but your attention completely shifts to the hot dog. And you're starting planning already the whole pattern of behaviors like, okay, when I'm finished talking to Reza and I'm finished with my poster presentation, I'm going to go and move and go and get my hot dog. And this is basically what we can sort of simulate in a lab. Of course, we don't do it in this type of, I mean, it would be lovely to equip all the grad students and postdocs with EEG recordings, but you cannot record the cerebellum, so we would only get frontal eye fields, which I'm not that interested in. Anywho, we did it in so we utilize the non-human primates, and this is the task. So what happens is that the monkey has to fixate, and then we use this called a land.c task. So it has to fixate on the middle of the screen, and there's a C that appears in one of the 4 locations of the screen. And then depending on if the C is open to the front, the side, or the bottom, the monkey has to make a saccade eye movements that follow up after there's a cue for making a movement. So how does it look? So what I showed you is actually how our very, very highly trained monkey, how fast they would do these trials. But of course, I gather that you have not seen the whole thing. So this is actually how the trials look like. So there's a fixation. There's a tiny cue that appears right next to the fixation point that will probe where the C is going to be. And you can see that if the C can appear on the left and the right, and if it appears on the left, on the right, it's going to be open to the top or the bottom. If it appears on the front, on the top or the bottom, it's going to be open to the left or to the right. So then there's, of course, a mask, because otherwise there's this visual after effect. So we have to mask it because otherwise the task would be too easy. There's a delay, and then the monkey is given a choice. And of course, this is we make it a little bit easier. So if the C was on the left or on the right, because it could be like both up or down, the monkey is only given these 2 choices. Otherwise, the task is quite difficult to learn. And as I said, the down or up C can point to left or right. So what we call it, we call it a gap direction. So the C has a gap, and we look at this gap directions. And then we also classify it as the up-down tasks, right? Or the left-right tasks, depending on the direction. And we trained 2 monkeys. I have to say that they were trained for several months, and they had been used for a pro and anti-saccade task before being trained on this cover attention task. So they knew what the fixation meant. They knew that if they make an eye movement, something good is going to happen. They had their own preference for different juices, by the way. This is also very important for non-human primate research. And I think straight off the bat, you can see that the monkey Mo was much better at this task than monkey Me. And so there was a difference in the performance. I should say that even for the monkey MI, the performance we would only consider like it trained to be higher than chance, and it was he was making considerably more errors, but overall his performance was higher than chance. But there was an error no matter how hard we trained. And of course, first we trained, and then we started recording. But I was also really curious about this difference in the behavior. And in all the previous studies that I cited, actually, the data from monkeys is very often pooled because for these simple tasks, they are trained to like 99, 98% accuracy. So there's very few error trials. Here, I'm very interested in behavioral variability and differences in the behavior. So when we started analyzing this data, we said, how about we try to understand why there's a difference between these 2 monkeys? Can we actually explain this difference in their behavior? So this is the work of Sue and Coy, who helped us with some of the analysis, and I'm going to mention her a little bit later when I'll be talking about the general linear encoding and then also a decoding model that she made to help us understand the activity that we find. But first, she just applied a quite simple logistic regression when we looked at what types of what were the things in the task that could predict the performance in the current trial. And here we thought of, like, for example, past error, past gap condition, current gap condition. What was the best predictor? And what you could see that, well, first, if you look at the weights of the current stimulus, so of the current gap direction, on the outcome of the current trial, you can see that well, Mo is of course much better, but also as you train it, this was, by the way, we looked at the sessions when they were also being recorded. So this is already in trained monkeys. This is the session 1 here is not the first training session. These are the sessions when they were already familiar with this. But if you train them, they still get their performance gets better, but actually not so much for the left right stimulus. So this is the green part. So the left right condition is something that monkey MI was never really good at. So he was better at chance, but he would always hover around like 60-70%. And then we looked at all these other predictions, like way to predict from the past trials and the past trial had no impact on monkey MO. So monkey Mo lived in the moment. He was really focused on what is happening right now and right there, and this would be the best predictor. Was really just he was paying attention to the stimulus. But what we saw here is that actually in monkey MI, he assigned basically less weight to the outcome, and you could see it that this was actually he was assigning more weight to the previous trial, and this was statistically significant. And I'm going to go back to this because this was quite quite interesting to us is why would he actually weigh in the outcome of the previous, why would he weigh in the outcome of the previous trial, and why could we see it in the regression model for the behavior? So we started recording these monkeys. As I said, we recorded from the lateral dentate nucleus using tungsten electrodes. So here you have an example of this electrophysiological recording. The C, this is this gap occurrence. Then there's of course a longer delay, and then there's the saccade. And here this is the reward period if the monkey would do a correct trial. So first of all, I have to tell you that this is a lot of conditions because you have all these down, up, right, and left trials. And then, of course, you also have either an error trial or a successful trial. So if you think of any type of data analysis, we actually started training these monkeys on 8 conditions. So we also wanted to have orthogonal, like 45-degree conditions. But very early on, we realized that because of the behavior, but also predominantly because of the data analysis, if we wanted to have the same cell recorded across all the trials, we have to simplify it, and we have to. So sometimes you have to make trade-offs with these types of recordings because you cannot hold on to the same cell for a very long period of time. So if you have 15 minutes with the cell, you're very happy. So what we noticed, and you can see it in here in these raster plots, is for many cells there was a very clear increase in the activity in the following the C period, and then a lot of them also had a response to the saccade. This is, by the way, an example of one cell. So this is why there's no shading, there's no confidence interval. So this would be an example of one cell. And because this is the cerebellum, and I just showed you these recordings from the 70s, when actually if you stimulate part of the cerebellum, there is directionality. Some of these cells would also have a preferred C direction and a preferred gap direction. And you could see here the numbers of cells that had, for example, there were some cells that had this preferred C direction, so up and down. There were 88 cells that actually had more of a more spread out gap direction. And this was in a stimulus, if you look at it in the preference in a stimulus window or in the saccade window. But what we saw very early on is that, first of all, I told you about the fact that the signals in the cortex were very heterogeneous. Well, here they were also quite heterogeneous because we saw that there were some cells that increased, so facilitated their responses, both in this gap, so instruction time, and also in the saccade period, but some of them also suppressed. And this was also true, by the way, for both monkeys. So it wasn't that there was one monkey had more suppressive cells and the other more facilitating. This bidirectional modulation in the dentate was true for both of them. And we also saw that the time of both either the peak or the throw, so here the throw of the response was actually a bit earlier for the suppressing cells, but that was mostly driven by monkey Mo, sorry. I was so used to working with them and using their names that sometimes I'm just going to slip and use the actual names of our monkeys. So this was largely driven by the monkey MO, but regardless, we found both facilitation and the depression and the suppressive cells. And then we thought, yes, so that was driven mostly by monkey MO. And then we thought, okay, this is again an example of a single cell. And again, this is a huge heterogeneity because we looked at, okay, so what now happens when you look at correct and incorrect trials? And then the responses of the cells sometimes would be similar and sometimes also would be different. So you have this at the descriptive level of statistics, we were a little bit stuck because there's only that much that you can say percentage of cells was facilitating, percentage of cells was pressing. What does it actually mean to the behavior? We asked the question, was there a difference in the activity when it comes to the onset of this modulation? So we did this type of analysis where we looked at the onset of the ramping. So the ramping is like when the cells start to increase their modulation. You can do it for both upwards and downwards. It doesn't really matter if it's facilitating. So this is facilitating cells, but you can also do it for the pressing cells. We did see that this onset of the modulation happened earlier in monkey mole, which maybe could also, to some extent, give us some hints why his behavior was better, because the immediately when the C came up, we could see that the neurons there started to modulate. But I think that we're a little bit stuck in how to explain it. And this is where we turned to Suen, and we asked her to do a more in-depth analysis. So we asked Suen, now she's a PI at Janelia, but she worked when I was a postdoc in Sam's lab. She was a postdoc at Princeton as well, and with David Tank and Carlos Brody, and she helped us to analyze this data. So we asked this question: what task variables can we actually detect? What task variables are embedded in these firing patterns? And for that, she modified a generalized linear model from the Pillow lab. So that's also a lab at Princeton. So everybody was very close to each other, so we could work together and really brainstorm about this data. And she used this generalized linear models to look at the encoding of the recorded cerebral nuclei cells. I have to say that we, of course, did some data pruning before we did this. So there were some inclusion criteria, like, for example, we had to have at least I think it was 20 trials per given condition in order to say anything about the activity. So that already dropped some of the cells that we showed you in the beginning. Nevertheless, we had enough to cells of both. And when I say all conditions, remember that we also want to have an error trial and also a positive trial. So it's not just 4 different types of trials, but it's 8. And first of all, what we saw is that we calculated the modulation index, and the modulation index is basically just shows you a score of how much trial types influence neural responses. So is there an influence of the trial type on the neural responses? And you could see that this is stronger. This modulation index is stronger in monkey MO that had a better performance for basically all different variables. So for the trial modulation index for the gap, for the past saccade, but also gap and the saccade, this was stronger in monkey ML. So on average, there was a stronger modulation in this monkey in the recorded cells. When we looked at what type of variables are encoded, I think I was very super happy when I saw this result. I think this was back in the day, so you have to understand that I think now everybody's like, of course, the cerebellum is highly heterogeneous. Dentate encodes everything. But when we started doing these recordings, this was before David Hetzfeld's paper came out. This was before all these big papers of reward encoding came out. So we were quite surprised and also quite happy to see how highly heterogeneous this group is. So we found that when you use this, when you make this model, we saw that there were a lot of different significant dependencies depending on the various cell types. So there were cells that had a significant modulation for gap and saccade that was actually very comparable between both monkeys, as was something that we already predicted, which would be a pure saccade cell. So there was also a fraction of cells that had a pure saccade response. There was also a mixture that had pure gap. And then there was a wide category of what we called like miscellaneous. And this miscellaneous recorded simultaneous different trial types. So for example, let's say that the one that says category miscellaneous 3 category, it would be simultaneous trial type dependencies on gap, past trial gap, and past trial saccade condition. So this would be multiple conditions. So you can see that roughly half of the cells in monkey MI and I would say a third, a little bit more than a third in MO have a gap and saccade or a saccade cell. And there was quite a lot that had several of these different categories. And here you can see these that it's not just we looked at what type of variables are embedded, but also over what period of time, right? So when you align it to different events, and then we looked at the correct trials and also at error trials. Interestingly, there would be, for a lot of cells, there would also be a very sharp response at the error trial. So this is something that we didn't really go into more details about, but you could see this error trial, this error response. And of course, when there's an error response, you have to think about the fact that the reward is also not given. Oh. Okay. So then we went. So after we did the encoding model, and we looked at what task variables are embedded in these firing patterns. The second was what actually information can be extracted. So what is the decoding model? And here, this I think aligned very nice with our with the observed behavior. And I think that what I really liked about this analysis is that we didn't put all these cells together. We always analyzed the 2 monkeys separately because of their different performances. And you can see here that cells with significant outcome were actually higher in the monkey MI. And you would say, huh, but he's a poorer performer. But that means that he made way more errors. So you have more cells that made more errors. So of course, then the decoding power is going to be better in this monkey, because for Moshe, he very often just had 95%, right? So there was not that many trials that were error trials. But when we looked at the cells with the significant gap decoding, that was higher in Moshe. And that, of course, is also relevant here because he performed better. If we looked at this past outcome, so this is not past gap, but past outcome. So whether there was an error trial or not an error trial, we can see this significance in Mickey. So it's almost like, his cerebellum was still stuck reminiscing in the past rather than focusing on what is currently happening. So the past outcome was still being able to be decoded from the dentate activity, but not in Moshe. And this was interesting because, again, if you look back at the behavioral model that we did, we could see this is something that brings us back to this behavioral model, where we could see the weight of the previous trial, so the N-1 trial, that we could see with the linear regression that predicted the response in monkey MO. So finally, what we also did is towards the end of the recording session, and I have to say, with some sadness, is that we wanted to actually keep on recording. There were still more cells that we wanted to do, but actually COVID came about, and we were sort of forced to stop the experiments. And then because one of our monkeys lost one of the screws, we decided that in the pedestal, we decided that we really couldn't risk anything happening to our subjects. So we decided to terminate these experiments once we had this experimental pool. Because we, of course, then also wanted to do with the reward with like, for the correct trial, withdraw the reward omission, random reward, but we just didn't have time to also include that in the setup. So what we did instead is at the last experimental days, we injected the tracer into the recording location. I can already tell you that if I had mouse injections like that, I would redo this. But of course, as you know, and for those of us who work with non-human primates, every non-human primate is extremely precious. So the recording, so this, if you see here that you have the dentate, they were a little bit on the border of the dentate. And also in Mickey, we think that there was an air bubble, unfortunately trapped. So the injection is smaller in Mickey than in Moshe. So with those caveats, we still looked at the retrograde labeling, and we found retrograde labeling in the inferior olive, as we expected. We particularly in the principal olive, you can see here we really cannot put them on the same scale, because if you look at the number of cells, you can see that this is much larger in Mickey, because you can also appreciate how much bigger this, how denser this injection is. But nevertheless, we were really looking at also the overlap between the two monkeys. And we also saw a retrograde labeling in the pons, predominantly in the medial pontine nuclei for both Mickey and Moshe. We also looked, of course, at the cortex, and this is getting back to this very beginning of the lecture when we saw that indeed we were recording the dentate nuclei cells that come from this very lateral part of the hemisphere, from the crus, some part of the crus 1 and the crus 2 regions. And you can see also, because the injection was so much smaller in MI, you can see that there is this overlapping region. So we think that most of the cells that we are actually recorded come from, receive the projections from predominantly this part of the cerebellum, where you have this overlap between the blue and the pink areas. CTB is not entirely retrograde, so there was also some anterograde labeling, and anterograde labeling there does not label cells, but rather processes. So here you can see the axonal terminals that we found in the thalamus, predominantly in ventral lateral thalamic nuclei, but also in pulvinar. We also found it in other parts, like higher in the brainstem. So, for example, yesterday there was a talk from someone who studies zona incerta, and I was like, oh, we also found projections to zona incerta from dentate. So that was, to me, quite interesting. We did not find labeling in ventral tegmental area. And I know this is something that we can discuss, because of course there's a lot of interest between the connections, between the cerebellum, direct connections between the cerebellar nuclei and ventral NVTA. There are some studies that show that's monosynaptic. There are some studies that show that it's disynaptic. we did not find any projections to the VTA, but this is, of course, with the caveat that this injections was not filling the whole dentate. So I just wanted to go back and finish this, that I think what is interesting here is that what is actually the output of this cerebellum? So what is this actually doing? And I think that this is very going way back again to Peter, who, by the way, also gave us a lot of help. I should say that I'm really grateful to Peter for giving us a lot of help with the perfusions of our monkeys and with this beautiful histology that we did, giving us a lot of tips. Where you say that the cerebellar output, he postulated the cerebellar output might provide the basal ganglia with this information to distinguish what kind of, so evaluation of the competing action candidates. So am I now going to go left or right? And before this action occurs, the cerebellum is saying, yes, I should do this, or no, I should do something else. So in summary, we show this high heterogeneity within within several the individual cell level. There's this multimodal integration at the cerebellar output stage, because many of these cells encoded both sensory and motor information. They modulated their activity throughout the whole task. So there were many cells that started modulating very early on and finished at the when the reward or an error was given. We showed that neuroactivity during this delay period to some extent can predict behavioral performance. And I really think that these findings support the important position of the dentate nucleus, both when we think about cerebellar modeling, but also if we think of potential targeting of like TMS stimulation or deep brain stimulation. And I just want to leave, before the acknowledgements, I just want to leave the fact that there are still remaining questions. This was actually a question, if you look at the eLife paper that we received from one of the reviewers, very fair question about causation. So is this part of the cerebellum even involved in this task? We were not able to perform the musimal experiments that we planned because of COVID and because in MI it was not possible. So this is still an open question. We did do one musimal injection, successful musimal injection in MO, and we did see a 10% reduction in his performance. So we did see a performance reduction, and this was significant because he was very highly performing. We don't know what happens during initial learning because we started recording when the monkeys were already trained. I would be also really interested in this interaction between dentate nucleus and frontal eye field. So if I had a monkey lab, I would be doing dual recordings during this task. I would be very interested in how the visual cortex and the frontal eye field in the frontal lobes modulates this behavior. And then I just want to leave you with this question about the theory of mind, right? Like we really have to discover that attention is such an human task, but I think it's a task that all mammals have to do, because you have to be busy with one thing, but you're already planning for more things in the future, and sometimes things that just appear in the periphery. So this is something that I think with these types of complex tasks, and I think with novel types of recordings, like for example what Reza is doing in your lab, maybe we can answer it. And with that, I just want to also just say that none of this would happen without Chris, but predominantly through a wonderful PhD student, Nico, who unfortunately left academia, but I'm still in contact with him. Also Peter, another PhD student, Bas, who did the histology, and Suan, who did the modeling. Thank you.
Thank you, Aleksandra. Wonderful. If you guys have anybody else question, please just go ahead and raise your hand. I'm happy to call on you. It looks like some of the students in my lab have a question. Go ahead.
You guys are muted.
Thank you so much for your talk today. I wanted to ask, there was a 2009 work by Young and Lisberger, which sort of touched upon a point from this work, which is that if you have a reactive task, then there's going to be perhaps more attention in non-human primates on the history. So, you know, saccade input -1 if this is in saccade. or in Smith pursuit. And for humans, there seems to be a focus on the current saccade, or all of the criteria related to the current, so error or reward that was received. I'm wondering if you also found this sort of connection, or can speak to this connection, that perhaps the more cognitively demanding a task, the more you're focused on the current circumstances versus the past.
Well, so I think that this is why in the beginning, we were very frustrated that the monkeys have different levels of performance. And I think it was really like a blessing in disguise, because you could see that here you actually had two monkeys with different behavior. One that really lived in the moment and that therefore was performing very well, even though the task demands were exactly the same, right? So it wasn't like that he got an easier version of this, versus the other monkey that still had this past result lingering on in its in the behavioral model, but also you could see it decoded in the neural activity. So the neural activity would still reflect the previous task. So I don't think that, so I here would argue that there's not an inherent difference between, of course, this is N of 2, by the way, so I don't know if I can generalize, but I would say that the monkeys, at least MO, was perfectly capable of I would say, the human level performance, and just paying attention to the current task, even though this is a very cognitively demanding task.
Let's go to Stanford and Jennifer's lab.
Hi, Aleksandra. So nice to see you.
Nice seeing you.
I've been thinking a lot lately about like specific behavioral tasks and what they tell us. And I was thinking about this as you talked about the mouse and choosing the social task that's like choosing between the object and the mouse. And there are many things that are different about a mouse versus an object, including just that one is more multi-sensory, one is moving, one is more sort of unpredictable, so there's lots of differences. And so I've thought many times as I see these tasks, like, what is it about the social behavior? Is it really explicitly the social part that causes a performance difference, or might there be these other elements that are not necessarily social but common to other tasks? So that and then I was thinking about it again, where you described the saccade, the C task, as like more cognitively demanding, but then it also has an attentional component and it has a competing action component, as you described. And so do you think there's a practical way to tease these apart? Like, what is is it attention switching? Because I was thinking about Eric Porchain's old work about how attention switching is slower, and might it really just be that and not the cognitive demand per se? Like, maybe a complex task is fine, but just it needs more time to switch attention. I mean, how do we go after these things, especially with these more complex tasks?
Very difficult question to unpack in like a time frame, but I have to say, so I think it was a really nice coincidence that yesterday I attended a lecture from Alexander Heimel, who doesn't really study the cerebellum at all, but he studies zona incerta and superior colliculus when it comes to novelty seeking and quote unquote curiosity. And I was really interested because of these projections that we found to zona incerta, because he and others, there were several Nature Neuroscience publications that show that, for example, zona incerta does this, has this drive to for novelty seeking. So if you stimulate it, the mouse is always going to choose a novel object. So I think, for example, for the social behavior, to get back to the social behavior, this is why I use the 3-chamber task in my lab. But this is why I always say it's autism-like. And it is a sociability task, but is this really truly social, or is this more exploratory novelty, curiosity drive, right? So that is something that I find very, very difficult to disentangle. This is why in my lab, for example, I started several years back to also use this live mouse tracker, where I look at sociability in groups of mice that are studied over long periods of time. And then I look at group dynamics like forming a nest, breaking a nest, which mouse is following which, etc. Because I feel that then we can say, okay, can I find this component, what I see in the social chamber during this 10-minute task, back in the behavior, in this innate behavior of the mouse that I can now follow for several hours over several days. So I think if we go back to the brain regions, I really think network is the only way to go. So I don't think that dentate is something that's going to be the answer. I think that these subcortical regions and studying the network dynamics between the cerebellum, between zona and certa colliculus, very interesting work. That is the way to go systems neuroscience wise. And then when it comes to behavior, I'm a big fan of, for mouse work, trying to disentangle it by looking at different behaviors. So
oh yeah, and I mean, you I think have the record for that in that Eli's paper that you did, right? Like that was really, yeah.
Yeah, and so I think multitask domains, and then doing some type of dimensional interaction. And my goal would be, for my lab also to do then recordings throughout different tasks. That would be, of course, a dream.
Well, let's go to MIT and Gabe.
Yeah, hi. Thanks for the really nice talk. So I have maybe a vague question, but a lot of previous work on spatial attention tests like these shows that they're in visual cortex, there's a spatially selective sort of gain input that leads to enhanced processing at a specific location. And that, when you stimulate FEF, you can, sort of mimic that type of effect. And so I just wonder, if you can help me connect the dots between how the type of activity you see in dentate relates to that type of model or thinking. Like how do you go for, are you thinking that dentate is influencing the attentional modulation, or is just reflecting that? Yeah, maybe just help me connect the dots a little bit.
So it would have been great if we were able to perform these musimal experiments, because then we would see how much of this is actually driving it. But from the work that I previously showed, for example, with lesion studies, right? And there's one experiment where we showed that there was a reduction. We didn't show it, but I can tell you that we have data when it was a reduction, but it was only N of 1. I don't think that this is something that is really absolutely necessary for executing this behavior. I think that what it helps is so in this last part that I showed from the paper from Peter Strick, but also if you look at Tanaka's work, that what I think is the dentate is doing in this task is helping to make the decision. So it's basically getting an inference copy of what is happening. It's processing it, and then it's making this decision. Yes, I have to go left and right. This is where I should make a saccade. And you can make that saccade without it, but it's going to be less accurate. You're going to make more errors, so your performance is going to drop. That's how I see it. But I don't think that this part of the cerebellum is necessary for purely the execution of this task. And I think that there's, so I had a lot of discussions with Peter Roosman, because he predominantly records in visual cortex and also in the frontal eye field. So we were also discussing this whole disinhibition and inhibition, right? Of when you look from the visual cortex to the frontal eye fields. And I think that this part of the circuits from cerebellum then to basal ganglia, I really don't care if it's disynaptic or trisynaptic or whatever, or monosynaptic, but I think that part would is important. So this sort of selection of what to do.
Yeah, that's really interesting. Just to follow up on that, I was thinking many of the neurons in the frontal eye field, motor cortex, at least in mice, during these delayed tasks, show ramping activity, certainly in non-human primates as well. And in those animals, during the deliberation period, damage or infusion of some antagonist in the cerebellum or optogenetic silencing disrupts that ramping activity. And in your task, you have to maintain the memory of the gap direction during that period, and that sustaining activity elsewhere through some loop that involves the cerebellum seems like some likely pathway by which that memory is maintained. I wonder if that that connects to the way you're thinking.
Yes, definitely. I was even thinking that you could sort of maybe recreate this type of task with the mouse, but then using whiskers, right? Like whisker stimulation, because I think that would be way more salient and then have it. And this is, of course, work that Sam has published a while back, where and Marlies Ostland also had a paper on bioarchive when she recorded from the cerebellum during these different puffs. So this, but that only reflects, that part of the task only reflects the working memory. So like accumulation of evidence and then the ramping. Here we also have this break where the monkey has a delay period and then it has to make a choice. So I was wondering whether we could recreate something like that and then tease out. So first do multi-electrode recordings, but then indeed inactivate different parts of the circuits. But this ramping activity, I'm a very big fan of rate coding. I'm very, actually, I don't know if I should say this out loud because there's probably some people here that are all about synchrony. But I think that in the end, a lot of the things that happen downstream is by integration of different parts of the rate coding and differences in the rate coding between the different neurons and tasks. So that would be, this is where I'm, if I would have to make a bet, that's where my bet is on.
Any other questions for Aleksandra? Thank you, everyone. Thank you, Aleksandra. It's great to see you. Wonderful. I hope
Thank
to see you
you.
soon.
Thank you so much.
Thank you.
Yeah, I will get on the other link, right? Yes, thank
See
you.
you soon.
Bye.